Hello, bachcho. So let's read the question. The question says, A. How does a gene therapy involving direct modification of the cells in order to achieve a therapeutic goal is used in the treatment of ADA deficiency? Explain. Second part of the question is, a host cell must be made competent before it is able to receive any rDNA justification. So the key concept for this question is, first is the gene therapy, and second is competence of cells in rDNA technology. ओके, सबसे पहले जो जीन थेरेपी होता है, इट इज बेसिकली a collection of methods. It is a collection of methods that allows a correction of a gene defect that has been diagnosed in a child or an embryo. So either in a child or in an embryo, we can do gene therapy, gene modification. How it is done is basically जो correction होता है, correction, it involves the delivery of the gene or of a normal gene. It involves the delivery of a normal gene into the individual or the embryo जिसमें भी gene therapy देना है, or the embryo to take over the function of the defective gene and compensate for it, ताकि वो डिफेक्टिव जीन का उसको टेक ओवर करके कॉम्पनसेट कर दे. सो बेसिकली इन 1990 एक 4 year old गर्ल को this therapy was given, or the gene therapy was given to treat adenosine deaminase deficiency या ADA deficiency. प्रोसेस में lymphocytes from the blood, they are taken and grown in a culture outside the body. उनको सेल कल्चर में ग्रो किया जाता है outside the body, and a functional cDNA, a functional cDNA, which is by using a retroviral vector, it is then introduced into the cells in culture, introduced into the lymphocytes. And then they are again returned or injected into the patient. सो इस तरीके से द लिम्फोसाइट्स आर इंट्रोड्यूस्ड इनटू द पेशेंट, बट हाउएवर ये लिम्फोसाइट्स जो हैं ये इमॉर्टल नहीं होते, दे आर नॉट इमॉर्टल. सो द पेशेंट हैज टू गेट दिस थेरेपी डन आफ्टर अ वाइल. इसीलिए इस थेरेपी को जब एम्ब्रायो में करते हैं, इट बिकम्स अ परमानेंट क्योर. कमिंग टू द नेक्स्ट पार्ट ऑफ द क्वेश्चन, जो है एक सेल को कॉम्पिटेंट कैसे करवाया जाता है सो दैट इट टेक्स अप द रिकॉम्बिनेंट डीएनए. So basically competence इस सेल को इसलिए दिया जाता है because DNA is a hydrophilic molecule. DNA is a hydrophilic molecule and it cannot pass through. It can it cannot pass through the lipid bilayer because lipid bilayer hydrophobic होता है. Cell, it has to be made competent to take up the recombinant DNA. So this is basically done by treating the cells with a specific concentration of the divalent. जो डाइवैलेंट केटायंस होते हैं उनके स्पेसिफिक कंसंट्रेशन का एक सॉल्यूशन में सेल्स को ट्रीट करा जाता है. फॉर एग्जांपल देयर इज कैल्शियम आयन. दिस बेसिकली इंक्रीजेस द एफिशिएंसी. इट इंक्रीजेस द एफिशिएंसी with which DNA enters through the pores in the cell walls of the bacteria. So इसके बाद recombinant DNA जो होता है या rDNA जो होता है, rDNA is the recombinant DNA. They are forced into the cells by incubating them with the rDNA on ice. इसके बाद they are again, it is this step is followed by briefly treating the bacterial cells at 42 degrees Celsius, जिसको हीट शॉक बोला जाता है, हीट शॉक ट्रीटमेंट. And then again putting them on ice. So coming back to the question, so the final answer for this question is that gene therapy जो है, it directly involves the use of lymphocytes which are taken from the blood and they are cultured outside the body, where the functional cDNA of the enzyme which is absent in the original DNA, it is introduced and then it is returned or injected back to the patient. And competence बढ़ाने के लिए basically the cells are treated with a specific concentration of the divalent cations and then इसको आइस में इंक्यूबेट किया जाता है. आइस में इंक्यूबेट करने के बाद हीट शॉक ट्रीटमेंट दिया जाता है एंड आफ्टर द हीट शॉक ट्रीटमेंट दे आर पुट इनटू आइस अगेन. होप यू अंडरस्टूड वेल. बेस्ट ऑफ लक.